Welcome back, friends, and in this future video lectures we'll be discussing about Mendelian genetics problems and some of the very important Mendelian genetics problems, and they will be from the classical genetics as well as the non-Mendelian genetics, that means the exceptions of genetics, Mendelian genetics. Okay, now in this future and subsequent video lectures what we will be discussing is not actually the Mendelism or the topics. So before coming to this video lectures, you must understand what are the Mendelian genetics concepts, the basics of genetics, and also the non-Mendelian inheritance pattern. That means the codominance, incomplete dominance, epistasis, multiple allele, polygenic inheritance, and sex-linked inheritance, and all those things. Okay, now if you don't know these things, you can search each of these individual topics in my website, in my YouTube channel, and you can find videos against it. More than one videos you can find. You can go through it, learn it first, then come to these problems. Now this type of problems are actually particularly designed for CSI and NET, because lot of problems from this genetics part are given. Now in these videos we'll be looking how to solve these genetics problems. Okay, now there are there's a common way of solving all these genetics approaches are simple by crosses. That means whenever we're talking about any kind of genetics problem, we based on cross. If it is a one characteristics cross, it will be a monohybrid. If it is two characteristic, it will be dihybrid. If it is of three characteristics, it will be a trihybrid. Normally in these questions, monohybrid cross is not given in CSI because it is too much easy, very much simple. So we'll be discussing from the dihybrid cross. Then they also give questions from trihybrid cross. Okay, now in the crosses you must understand the Punnett square, how to produce the Punnett square, and how to derive the genotype as well as phenotype ratios of the F1 generation, F2 generation, and further generations like that. Okay, now you can do this utilizing those Punnett squares. You can do this utilizing probability. Okay, now in these videos I am the fan of probability questions. So in this particular part I'll be teaching you how to solve these genetics problems with simple probability rule. Okay, now I am not going to take any theory class about that. So I simply start with examples, and during those examples, explaining those examples, I will be teaching you how to solve the questions, because I found it much more effective than simple telling the theory first, then all these things. Okay, so let's begin with questions, simple questions, so it will help you to understand how to solve them. Anyways, so let us first talk about dihybrid crosses and some of the examples of dihybrid cross. Very simple example, and we won't talk about monohybrid because it is too much easy, so we won't talk about it. Let me take this. Okay, so here, say this is the question. In peas, a gene for tall plant is caps T is the dominant over an allele for short plant, which is small t. So T, this is the question number 1. T denotes tall plant, small t denotes short plant for P. Okay, and the gene for smooth peas is caps S, which is also dominant, and for wrinkle peas is small s. So caps S on the other hand, so let us take it with different color. If we are having colors, we are having lot of colors, so let us change the color here. So this is smooth, and say this is wrinkled. Okay, so we are having this 4 type of things. So we are having 2 characters and 2 alleles for each of the characters, so 4 different alleles here. The genes are not linked. Okay, so as this term says, the genes are not linked, simply tells you that this whole genetic pattern will be of Mendelian genetics. Okay, if the genes are linked, then that will be from the non-Mendelian genetics. So if they are telling these genes are not linked, that means you are dealing with Mendelian genetics. So the ratios will be depending upon the Mendelian style. And now they question you, they calculate both phenotypic as well as genotypic ratios for the result of each of the following. So phenotypic and genotypic of that. So say they cross, they say the cross between TTSS with TTSS. So both of them are heterozygous in nature. Okay, now let us talk. This is the cross. Okay, now we can do this. We can do this cross simply by producing the Punnett square. So Punnett square 16 things will be coming. So let us do this for both the case. So for the first question, let us understand from both the perspectives how to solve this question. Okay, so what is the genotypic ratio as well as phenotypic ratio? They won't tell us the traits, right? So what will be the traits? Now if you are getting this, what will be the traits? So simple combinations of these four things, it will giving us the traits. So so say the traits will be here, say tall. So let us write down the traits: tall and smooth, tall wrinkled, short smooth, and short, not like short wrinkled. These are the four phenotypic differences that we can get. Now I am telling you one thing, this is very much important. In all this type of crosses, what is very important is this phenotypic ratios. Because genotypic, you can get genotypic ratios. With phenotypic ratios, most of the question will ask you for the phenotypic ratio. Because most of the dihybrid cross must have the genotypic ratio, most of them are the similar. So they usually looking for this phenotypic ratio. So all these things are among phenotypes. Okay, phenotypes of this cross. Okay, of this plant actually. Now our approach will be different, right? It's a probability approach. Now in this probability approach, how we are going to solve the question is, we will take each of the character, each of the trait, and we'll be looking. So here there are two traits involved. One is the length of the plant, so it is a tall or short. Another one is the type of seed, smooth or wrinkled. So here we'll be taking each of them individually. So let us first take the length of the plant. So it's a tall or short. So if we simply make cross between these two T's only, so you are only taking, say, these two things. We are only taking these two things. So what will be it? So let us simply assume. So in this case I am just figuring it out, but as you are going to do the maths, you will be knowing that what is the outcome of this simple monohybrid cross, because it will be a monohybrid, right? It's a monohybrid cross. So monohybrid cross you can just figure out in your brain. You should be able to figure it out in your brain. Now I am doing this because I am doing this for the first time in front of you. So let us do it. So this is the monohybrid cross. So this will be TT, caps T small t, caps T small t, and again small t small t. This is a monohybrid cross. If we take only these two and cross between themselves, very very important, we will take this and cross it. The second time we will cross these two things. Okay, this is. On the other hand, see we are crossing this S with, so caps S, small s, small s, small s. Okay, so what are the ratios for both the cases? The ratios are, again I'm telling you, the ratios for T, it will be caps T small t, so heterozygous, and again homozygous recessive. So homozygous dominant, what is the probability of homozygous dominant? It is, say, the total offsprings are 4, right? So and caps T is only 1, so it is 1 by 4. This is the ratio. Getting it or not? Say for the next thing, heterozygous, we are having 4 offsprings. How many heterozygous? 1, 2. So we are having 2 out of 4. And on the other hand, again this is 1 out of 4. Getting it? Similarly, for this one also, we are having caps S, small s, small s. For caps S, we are again having 4 offsprings totally. So then caps S is only 1, so 1 out of 4. Caps S small s is 2, so it's 2 out of 4. and again smallest is also 1 out of 4. So these are the offsprings we can get. Okay, now what possible difference these things can make to us? Now here we'll be looking for the phenotypes. So let us write down the phenotypes for here. Say we are talking about, say, from this point of view. Okay, what we'll be figuring it out. So we are going to step by step approach. The first step is to create monohybrid cross. So let me take another color. The very first beginning is to create the monohybrid cross. Monohybrid cross, and we have done it successfully. We have done the monohybrid cross. Now the second step of solving this via probability is to get the ratio of phenotype in monohybrid state. Get the ratio of phenotypes in the monohybrid state. That means the ratio of tall plant is to short plant, and also the ratio of smooth peas is to wrinkled peas. Are you getting it or not? Just go back, rewind the video and look at it. First step, monohybrid cross, you can get the monohybrid cross ratio. Second thing, you have to get the ratio for each of this monohybrid cross. Okay, so this is the second step. Ratio, to get the ratio of monohybrid cross. Now if you are having this, so what will be the answer for that? So the ratio tall is to short. So let us find it out. This is tall. This is also tall. So 3 are tall and this is short. So 1 is short. So the ratio tall is to short is 3:1. Okay. And ratio of smooth is to wrinkled. What are the ratio? Again, this is, say this is smooth. This is also smooth because smooth is the dominant character. So heterozygous would be also smooth. So it is smooth, this is smooth. So 3 is smooth, 1 is wrinkled. So again, smooth is to wrinkled, and the ratio will be 3:1. Get it? Now, how can we derive the phenotypes, like this fourth type of phenotype as well as the genotype? Let us find out the phenotypes first. This is called cross multiplication. Now, just whatever I am going to do, look at it very carefully. Using these 4, we can produce 4 combinations. Like first one, tall, smooth, crossing these 2, tall, smooth. So if we multiplying tall, smooth, we are getting tall, smooth. Okay. So tall, smooth, we similarly multiply these 2. So 3 into 3, 9. Okay. Is 2. Smooth, now let us multiply it like that. So we are fixing smooth here. Multiply it with tall first, multiply it with short. So short, smooth. Okay, I'm not writing in detail. Short, smooth. So similarly multiplication, 3 into 1, 3 is 2. Getting it? Now say take wrinkled. So wrinkled, tall. So it will be tall, wrinkled. It will be this 3 into 1, 3. Okay. And the final one, short, wrinkled, right? This one, 1 into 1, 1. So what is the phenotypic ratio here? 9:3:3:1. Now this is a crucial step, the most important step of all. And for doing this, this is the actual step you are going to perform in your exercise book. Whenever you are seeing a question like that, all of these above things must undergo in your mind. You shouldn't write this because you don't need actually. I'll giving you some important trick, important ideas that can help you to just figure this out by looking at this crosses. Until this second step. This is the last step that you must do in your writing. Okay, so simply what we are doing, we are just multiply things. So we just fix smooth. So now take smooth in horizontally, then corner. So tall into smooth, short into smooth, wrinkle, tall into wrinkle, short with the wrinkle. Okay, similarly we will multiply these things. Smooth into tall, smooth into tall. So 3 into 3, 9. Smooth into short, smooth into short. 3 into 1, 3. So wrinkled into tall, again here also wrinkled into tall, 3 into 1, 3. And wrinkled into short, here will be wrinkled into short, 1 into 1, 1. So this is how you can solve this. Now if you want to solve the same question with drawing a Punnett square, it will be much more hatred. You know why? Because in the Punnett square you will be getting 16 different type of genotypes. If you want to, you have to find which one expressing which phenotype. So you have to find those things out, and it will be much more tedious. What happens sometimes you bypass some of the phenotypes. Sometimes you won't just count it. It will be very much hatred. And for the tri-hybrid cross, if you want to do a tri-hybrid cross, it will be 64 squares. So it will be much more hatred in those cases. So for these purposes, if you follow this probability rule, it will help you to go these problems very fast. In the future videos, we are going to see how much faster you can calculate these frequencies. Okay, in the future videos I'm not going to do these things. I'm just going to do this in your mind and tell you how to calculate that in your mind. Then you can simply just do this step and you get the phenotypic ratios. Okay, so this is about the phenotypic ratios. Now how can we calculate the genotypic ratios? Okay, now for the genotypic ratios, it is much more easier because you already calculated out the genotypic ratios here. Okay, so let us go to explain the genotypic ratios here. Say I just take genotypes here. Okay, this is clear. Now let us go for the genotypic ratio. So the genotypic ratio here, caps T is 1. So let us write it like that. Into caps T small t, it is 2. Again small t 1. On the other hand, for the other character, so we are doing for the probabilities simply separating that type of characters. So if they give us 3 characters, we'll be doing this. We'll separate this 3 times. Simple as that. So let us say caps S is 1, caps S small s is 2, and again small s is 1. This is what we get. This is what we get. Now how can we calculate? Similarly, just crossing them. So again caps, so just multiplying things. So number is 1. So again say we just making this one constant, and this, now then this, then this. We'll multiply things like that. Then multiply caps t small t, 2 into 1 2. Again caps small t 1 into 1 1. Okay, now let us take this one. So caps t small s, 2 into 1 2. Then this one caps small s, caps t small t, 2 into 2 4. Then this one caps small s, small t, 2 into 1 2. Then this last one we remain this only. So this with this first. So caps t small s 1 into 1 1. Caps t small s 1 into 2 2. Caps small t small s 1 into 1 1. So this is what we call the genotypic ratio. So for the phenotype we only hold on to the phenotype and do it, and for the genotype we will write down whole things. That's so this is the thing, we can get it just very fast. In the very first step we'll figure these things out. So from this step, for genotype we'll just place this thing and make the multiplication and get the result. For the phenotype we'll just figure out the ratios, then we'll multiply it. That's the only difference. Now here say what is the ratio? 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Okay, so this is a huge ratio, right? So the ratio here is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So this is the genotypic ratio, and 9 is to 3 is to 3 is to 1 is the phenotypic ratio. So this is the genotypic ratio, right? And this is the phenotypic ratio for this cross. Now you you're probably thinking that for the answer, for getting this answer, it will take 15 or 20-minute discussion. But it's actually to show you for the discussion. When you start doing these things in your mind, it will, believe me, it will come down to 1 to 2 minutes, maximum of 2 minutes, not more than 2 minutes. You can answer these questions, and for CSI and NET, these questions will demand you giving you 4 marks. For 4 marks, if you utilize only 1 to 2 minutes, it will be very, very advantageous for you. Okay, so let us find out much more frequencies. So this is the first type of cross we have dealt with. Now I'm giving you another problem, and I'm going to tell you how to figure these things out pretty fast. Okay, now a very quick tip for you is that for this kind of crosses, for the monohybrid, for this dihybrid crosses, how to quickly figure out the frequencies, how to quickly figure out the ratios for monohybrids for the first time. So let us say in this case what we are getting it caps T small t into caps T small t. So if you take only these two, we are getting a cross like that, because in the whatever cross we are given, it's a dihybrid or trihybrid, we are separating the traits apart. We will do these crosses as a monohybrid in separately, then we'll just combine them, right? So in this case, if two, remember this, if two heterozygous are crossed with each other, we can have only three. Any case, for any monohybrid cross, we can have three possibilities. This is clear to you or not? You can have homozygous dominant, you can have homozygous recessive, and you can have heterozygous. Heterozygous is only one type. So in any kind of monohybrid cross, you can have three possibilities: homozygous dominant, which is caps T, homozygous recessive, small t, and a heterozygous caps T small t. This is clear. Now if you are crossing two heterozygous traits, in those cases always the ratio is always heterozygous is the highest. Remember, if you crossing two heterozygous, always heterozygous is the highest. So the heterozygous is above 50%, so it is 50% always, and the rest of the things are 25-25. So the ratio always, if you cross a monohybrid, always the ratio is 1:2:1. This thing keep in your mind. So whenever you are looking that this is the first, so you're getting the question, you're looking at it and you're looking at that, yes, this is cross, dihybrid cross, separating these two traits out, T out and S out. Then you take caps T small t in cross. You just look that both of them are heterozygous. Simply it will tell you that the ratio is 1:2:1 because it's a heterozygous. Remember this. If it's a heterozygous cross, always the ratio is 1:2:1. Get it? Now if the cross is between two homozygous, if the cross is between two homozygous, if the cross is between two homozygous dominant, all the offsprings will be homozygous dominant. Sorry, if both of them are homozygous dominant, then all will be homozygous dominant. If both of them are homozygous recessive, all will be homozygous recessive. These two things are clear. If this we have discussed this, then it will be 1:2:1 ratio. If the cross is homozygous dominant, all offspring will be homozygous dominant. If it is between two homozygous recessive, all the offspring will be homozygous recessive. If it is both of them are heterozygous, then the ratio will be 1:2:1. And the fourth possibility, if one of them are homozygous, another one of them are heterozygous. Here lies the tricky part again. One is homozygous, another one is a heterozygous. What will be the ratio? So let us count. Caps T small t, so caps T small t. So let us look, telling you homozygous are 2 out of 4, heterozygous are 2 out of 4. So it's a 50-50. If one of them are homozygous, one of them are heterozygous, so the chance is 1:1. It's a 50-50 chance. Okay. Similarly, if you can tell, same thing. If one of them is homozygous recessive, another one is that. Simply, again, if these things also happen, then also say these things. So, similarly, again, homozygous one, heterozygous one. So, in any perspective, whether it is mating with both of them homozygous, offspring is 1:1, offspring, all of the offspring is homozygous. If it is recessive homozygous, offspring will be homozygous. If both are heterozygous, ratio will be 1:2:1. If one is homozygous, one is heterozygous, the ratio is 1:1. Print this in your mind. Write down in your notebook. This is the formula you need to learn. You need to place it in your mind to, if you want to solve these problems with probability. So whenever you're getting it, again, look at this problem. Again, capsa small. If you are looking at SS, so capsa small s with capsa small s. So both of them heterozygous. Promptly you think, what will be the ratios? 1:2:1. Remember, if two heterozygous cross, the ratio will be 1:2:1. Say, if I am giving you another cross, if I am telling you that one is this into this, if we telling this is the cross, then let us decide here. Okay, so write it here. If they give you a cross into this, if they are giving you a cross like that, now let us, can you tell me what will be the ratios just by looking it? Caps T is caps T small t. So one is homozygous. So here divide it. First divide it. Then one is homozygous, one is heterozygous. What will be the frequency? One homo and one hetero. 1:1. Yes, 50-50 frequency. Get it? For the second, so the results will be, this will be 2 out of 4, and caps T small t heterozygous, this is also will be 2 out of 4. Remember, right? Now in this case, S, both of them heterozygous. What will be the frequency? 1:2:1. So caps small s, small s. This will be 1:4. This will be 2/4. This will be 1/4. Looking at homo with hetero, answer will be 1:1. Hetero with hetero, answer will be 1:2:1. This is the most important part. Okay, by looking at it, whenever you start dividing things and by looking it, you should able to tell this by just looking at it. So just looking at it and figure it out, then everything will be easier. Just write this step, get the answers. Okay, now let us solve a problem about this. Now let us, okay. Delete it and let us start the same. So everything remains the same in this case, but what will vary? So delete it. Okay. Now the question they give you is the same thing, that cross characters remains the same. Simply the type of cross is changed. Say they tell you small t caps S small s is crossed with caps T small s small s. Okay, so they give you this question. Now quickly figure it out. Pause this video, look at this problem, try to solve it on your own. Okay, then we will discuss. Okay, now let us say, just first divide this. So let us take a T's first. So the cross is one homozygous, one heterozygous. What will be the ratio? 1:1. So the ratio is of one homozygous, one heterozygous. Both of them will be 2 out of 4. Get it? Okay. Second thing, take the S's. One is hetero, one is homo. So again, homozygous is to heterozygous ratios will be 1:1. So 2 out of 4, both the case. Okay. Just by looking at it, you can figure these things out. You figured it out. Now let us look at the traits. So what will be the traits in this case? tall is to short, again smooth is to wrinkled. We need to figure this out for the phenotypic ratios. So here how many talls? This is a tall. So 2 tall, these are shorts. So only 2 talls, 2 shorts. So the ratios are here 1:1. And also again wrinkled. This is smooth because smooth character is the dominant. So heterozygous will be smooth. So 2 smooth, 2 wrinkled. So the ratio is again 1:1. So ratio is again 1:1. So these are the ratios that we can get. So what are the phenotypic ratios from that? From that point of view, let us calculate the phenotypic ratios. Okay. So here tall into smooth, 1:, smooth is to short, 1. So whatever multiplication we can do, all these things are ratios are 1:1. So whatever multiplications are, it will be 1, right? 1:1, always 1. So the ratios for the phenotypes are 1:1:1:1. So now let us go for figure out the genotypic ratios. So genotypic ratios say caps T is 0, caps T small t is 2, small t is 2. On the other hand, caps S is 0, there is no caps S, then caps S small s is 2, small s is 2. So multiply them, again the multiplication rule. So caps T is 0. So whatever we are calculating it will be 0. So we will exclude this, we don't need to think about that. Take this one, this will be a 0. Caps T small s will be a 0. Then this one, caps T small t, caps S small s is 2 into 2, 4 is 2. This one, caps small t, caps S small s is 2 into 2, again 4. Then this will be also 0. Caps T small s it will be a 0. Then caps T small s 2 into 2, it will be a 4. Small t small s it will be a 4. So the ratio again for the genotype purpose 1:1:1:1. Okay. So for phenotype is also 1:1:1:1. Genotype is also 1:1:1:1. You need to write this genotype. You can tell that this is the genotype. So now you can get how fast you can just figure this thing out. This amount of time it will take. Just blink of second you can figure out these problems. Okay. Now in future videos we'll be looking more problems and we'll be learning to solve them using these techniques. Okay. So I hope this video will help you. Thank you.